interface is another layer that allows to run CellOrganizer on bridges. So every time that we run something here on CellOrganizer, on Galaxy, it will submit a job to the cluster, to the scheduler. It will run. During regular hours, there's some waiting time. There's a lot of computers, a lot of nodes that are a part of bridges, but you will submit it to a scheduler. There are a lot of people in line. You have to wait. But for the purpose of this tutorial, we had what we call a reservation, so you'll see your job start immediately. Okay, so there's no lag for today. A few words that I want to mention before I start this presentation. You'll see me or you'll hear me say the word framework a lot. A framework is a nuclear membrane and a cell membrane. So we call them the nucleus, the cell, the framework, and then whatever protein pattern that is we're talking about. The reason why we call it a framework is because the way CellOrganizer works is that you give it a list of models, but it will generate the framework from the first model in the list. And then it will populate it with the rest of the protein patterns in the rest of the list. Does that kind of make sense? So, okay, good. That way, I might need my charger. Okay, good. So the first thing we're going to do is that if you move to your right, you'll see something. You see this here where it says history? Okay, so history is similar to a workspace. So what we're going to do today is a series of exercises. Each exercise will have their own history and their own results. You can share your history with other members of the same instance. So technically speaking, we could share results if we want. So the first thing we're going to do is establish this history. So we're going to click here on unnamed history. I'm just going to call it exercise 1. And then click enter. And then I'm going to click this little tag here. Just for the purpose of matching things, I'm just going to call this train model. I'm going to call it classic. So what we're going to try to do right now is to train a simple model. Like I was telling you before, if you see that little page, I don't have it anymore. This page over here, I told you that we always have to pay attention to this table, okay? The information, besides the images that the system needs from you, it's dependent on the combination of model classes and types, okay? So if you want to train a diffeomorphic model of the cell, it will ask you specific questions about your images and so on. The model that we call the classic model, or at least I refer to as the classic model, is the Vesicle model. It's basically the cylindrical surface for the nuclear membrane, the ratio model for the cell membrane, and the GMM, the Gaussian mixture model for the vesicle. Now, ideally, if you were doing your own images, you would just select Get Data to upload some files from your computer. And then you get a menu like this, and very simple, and you would just click Choose Local File, and then you would select from your hard drive. You click Next, and it uploads these images into your workspace. What we're going to do today is we're going to get some images from the shared data. And if you click at you see at the top, right, you see that shared data? So I, as an admin, share with all of you the images that we use with CellOrganizer. So if you click here, you're going to see data libraries. Okay, and we're just going to click that, and it's going to take me to this menu. Is everyone there? Yeah? And then you'll see annotations, images, and models. And this is the information we shared with you. And I'm just going to click on images. And then the only image collection that I have is the Gila. So I'm going to click Gila. We're going to train a 3D model because that's the one that's kind of interesting. And so I know beforehand a little bit about these images. I know that LAMP is going to be the model that's going to be faster to train because it has the smaller number of objects. So I'm going to click here, and it's going to take me to process and raw. So I talked about it this morning that to CellOrganizer you can either submit images that are raw, and then there's a specific pipeline that will try to clean them up, remove some noise, do some simple pre-processing. What I cheated for the purpose of this tutorial, and I cleaned them up beforehand, so computation would be faster. So I'm going to click Process, and I'm going to see my list of images here. At the bottom, it's telling me that it's showing me only 15 images of 50. So I'm just going to click on the 15, and then I'm going to change this.
How are these processed?
What?
How are these processed?
We basically identify the largest object, remove background noise. We
Identify the large object, so is it like metadata or something?
no. It's just like regular, just image processing. We have algorithms for identifying the largest object. We do have some manual segmentation. Yeah, we do a couple of things to the image. We clean out, they do active 3D contour segmentation to get the object. So we have our own pipeline that we know that works on our images. That's the whole point. And then this is what we are confident are the objects for us. So I'm going to click 50. So I'm going to get them all. And now I'm going to see the whole list of images. And all I'm going to do here is click here next to name to select them all. And now you'll see here, you see this little button that says through history? I'm just going to click that button. And now I'm going to move it to exercise 1. See, select history, exercise 1.
But how do you select them all? Just like
Oh, here, see here, see here?
Oh, okay.
Name?
Yep.
But you need to make sure to have all 50 images there. If you don't have them, you have to like go through each page. Okay, so I'm going to click import, and it's going to import the images into my history. Okay, so I'm just going to wait a little bit. Is everyone doing the same thing? Is everyone lost? It will be kind of slow because there are like 20 of us doing it at the same time.
My one's already done.
It is-
With the class and type again.
What?
With the class and type that we use.
Class type?
Class, model class, and model.
Oh, you'll see in the menu. We're going to do a full vesicle, a full classic model. Yeah, we're just importing the images into the history.
Images did you do?
What?
3D, but which ones? All?
Lamp. Yeah, lamp 2. Now it's a very good question that you ask, because we're training the framework with the nucleus. It doesn't make sense to use the whole collection. But if I was trying to learn a framework model on HeLa cells, I could potentially use all of the protein patterns and then just ignore the protein and just train the framework. Oh wow, this is kind of slow, but this is working. And what this process is doing is basically bringing the images into your workspace. Click here in the green banner, and it will take you back to the history. Are we all here? Okay. So if everyone is here, all we need to do is click this check mark over here, and we need to click, and you'll see that you have this line over here. Says all, none, or for all selected. We're going to select them all. And for all selected, we're going to make a data set. Okay? Is everyone there? Are we on the same page? Okay, then just click, and it's going to ask you for a name. So here at the bottom, I'm going to say hide original elements. And I'm just going to call these images. You can call them whatever you want, but you know. And then create list.
Build dataset list.
Yes. And then you should see something like this. The important thing is at the bottom you should say a list of data sets. It's kind of confusing because for Galaxy, a file is a data set. But for us, a file is an image. So whenever you see the word data set, it just means an image. Okay, so we created that image. And what we're going to try to do is train a model. Okay, so let's see if we can do this correctly. So on the left, you have all the different options. And you have one tool for training. I'm just going to click training. It's going to expand. And I'm just going to click train a generative model. Now, I mentioned this morning that you could do a lot of things with CellOrganizer, but we only exposed some of the simpler options to the user through Galaxy. So like I said before, the important thing is the combination of models of classes and types. So the first thing I'm going to do is select the data set. So I'll click on dataset collection, and then you'll see 51, which corresponds to my images, the image set that I selected. I'm going to click that. It's going to ask me for the dimensionality. I'm going to say it's 3D because it's a 3D image collection. And then it's going to ask me for an index number. Now, these images that we have uploaded to Galaxy are OME TIFFs. So these are a multi-stack that are separated by an index. So in this case, I'm saying at index 4, you can find a mask for the images. At index 1, you will get the DNA channel. At index 2, you will get the cell channel. And then CellOrganizer can process those images and extract the right stack and put them in the right position in the model. And then here at the end, select the cellular components desired for modeling. We can either train a nuclear model, a framework model, or an all model, which is a way of saying nucleus, cell, and protein. And for now, we're only going to do framework because we want to do it relatively quickly. And provide a model name. I don't know. I'm going to call it MMBioS. Okay, you can call it whatever you want. Oh, MMBioS? No. MMBioS. Provide a default ID. I don't know. 1. Advanced. And this is important. Like I said before, the time it takes to train a model is highly dependent on the resolution, and it's depending on a downsampling factor. We can downsample the image to speed up processing. The information about the resolution of the image is kept at the metadata level of the image. So it will extract that information from the TIFF file. That's why it's not asking you. There is a little bit of overhead at the beginning when you're trying to train a model, because it will literally check all of the images in the list to verify that they all match up in terms of resolution. Okay? So that's why at the beginning it's a little bit slow to start. And then I'm going to downsample on the X and Y direction by a factor of 5 so that I can accelerate processing. And then I'm going to ask it to save intermediate results. So saving intermediate results is really good, especially for debugging. You can set the debug flag to true. And then what happens is that all of the intermediate steps that are saved by CellOrganizer are kept after the model has been trained. And then you have the options regarding to the nucleus, cell membrane, and protein. For nucleus, we talked about doing a 3D nuclear membrane cylindrical surface. So the nuclear model type is cylindrical surface. That's good.
Why the
Hmm?
Why the nucleus cylindrical? Why didn't it be more
Because it is an extension of the medial axis model, right? Is that how the cylindrical surface model works for the nucleus?
Yeah, it's our 3D version of the vector axis model.
Yeah. Yeah, I think for the 2D case we only feed a spline, which works pretty good.
The 2D you own the vector axis, and then
Yeah.
like for 3D you need the number of surfaces.
Now, the thing about this kind of model, the cylindrical surface models, is that it sort of creates like a smooth top layer and a smooth bottom layer, because it's incapable of measure like curves at the top of the nuclei. So you'll see them; they look kind of like cylinders. That's part of the cylindrical surface. But this is the very basic model. It works with the HeLa cells, but it's not necessarily efficient with all kinds of cell shapes. So we're going to select cylindrical surface, nuclear membrane, optional name. You can leave this. You can put whatever information you want. I'm just going to say nuclei. And then the same thing for the cell membrane. We're going to train a cell membrane ratio model. Oh, it's selected by default. Cell membrane ratio, provide name.
Which one is it? The cell membrane?
It's a cell membrane ratio model. And like I said before, the parameters are extracted from the images automatically based on this selection. So it requires very little input from the user at this level. There are more options, but we just kept them to default now for the and then it needs some information from you. What is the location of this protein? So this is a thing, an endosomal protein, so I'm just going to keep it on that.
What if it's unknown?
We put all. Okay? And all means that it could appear either. Because the issue about this is when you synthesize an image, you can force it to appear or not appear in whatever compartment you're familiar with.
Mm-hmm.
If you put it in all, and at the moment of training, there weren't any objects, say, in the nucleus, and the probability of finding an object in the nucleus is very limited, but it's non-zero. By selecting cytoplasm, you're guaranteeing that if an object gets synthesized inside the nuclei,
It is zero.
it gets removed.
Mm-hmm.
Okay? And then documentation, you can put whatever documents you want. So, hey, it's me. I'm not Iceberg. I'm Iceberg always. And then, I don't know, email. And we always ask you to document your models. It's a very good practice. So we know when do you generate them, what computer, all the extra information you want. It's always important. And then that's it. I'm going to click execute. And if I click execute, I'll see that to my right, I'll have this little box, hopefully. Very good.
Mm-hmm.
And that means that my job has been submitted. Okay? And now,
Why do we have two of them?
hmm?
Why do we have two jobs, 52 and 53?
What do you have two?
Yeah,
Two outputs, you mean?
yeah.
Oh, because one is the actual model file, and the other one is an HTML file that links all of the information generated by the training of the model. So by clicking that little report, it will give you some information about the model. You will be able to download the option structure used to train this model. It will download the temporary folder, and it will download another folder that contains all of the parameters that were extracted from each of the images. Generally speaking, if you run this in the desktop edition, you will see it locally, but it's the only way that we can provide, we can give the results back to you. The reason why it's embedded in an HTML file rather than one of these outputs is because there's nothing you could do in Galaxy with those outputs. You can just download them. The model file itself, yes, it's important. So is everyone, you should see them in yellow. Oh, good. Does everyone see them in yellow?
I have exclamation points.
Okay, you can refresh the page. If you see the exclamation points, you can refresh the page.
Okay, thanks. Yep, they're going.
Okay, so I wouldn't call it a problem, but here's the thing about Galaxy. You will not be able to get any feedback from the system until the job is done. Okay, so right now, because you see this yellow box, that means that it's running. Everything seems to be working properly, but because everything is running in the cluster, you will not get any feedback until the job is done and all of the files are returned. Okay? So let's do a little cheating to see we're doing things correctly. So I'm just connecting to the HPC, and if I do watch, what do we see? Okay, there we go. So these are all of us. Okay? So we successfully submitted all of our jobs to the cluster, and all of the jobs are running. That's what DR means. We have been running them for about 2 to 3 minutes.
Wait, how did you get this?
Oh, this is me. Because when you submit this, when you submit a job through Galaxy, at least for this instance, you're submitting your jobs as myself. You're using my allocation. Okay? So it looks like in the system that you're running my job. So I'm just going to take an example of any of these. I'm just going to copy this job ID. And I have a little There you go. So this is the output of one of us. I cannot say which one, but it's calculating cell codes, it's extracting statistics at each of the stacks in the 3D image, it's downsampling, it's extracting all of this information, and everything seems to be working properly. Okay? Unfortunately, there's a little bit of waiting when you do these things on Galaxy. And so we're going to go back. And I don't want you to just sit down and wait to see what it's really happening. So let's try to make some images, okay? So when this job is done, I'm going to get a model. But let's come back to that model like in 5 minutes or 10, whenever it's done. So let's try to grab another of the models that we have in the system. So now, wait. So I'm going to go back to this little gear. History options. I'm going to click on that history option, and I'm going to select Create New. And it will create another history. And I'm going to do the same thing as before, exercise 2. Boom. And now I have a second workspace. Now, let's try to grab some things from the share data. I'm going to click on share data, data libraries, but instead of grasping images, I'm going to grasp a model. Now, these models that we're sharing with you are the models that have been trained on this specific cell lines and that have been trained at a higher resolution. Okay? Always keep in mind that the good thing about CellOrganizer is that it does take some time to train these models, but you can share them with other users as well. So I'm going to go to Gila. I'm going to take 3D. And let's see, we're trying to synthesize lamp, train lamp, so why don't we get something else? Let's do MMBioS. I'm going to click on MMBioS, and then I'm going to go back and say to history. Are we all there? Am I going too fast?
I'm trying to get, how do we get the libraries again?
Okay, let me go from the beginning. So I created the history here. I'm going to go to share data,
Yeah.
data libraries,
Oh.
and then I'll see the 3 options. I'm going to select models,
Okay.
Gila, a 3D model,
MMBioS.
and MMBioS. Now, to history. Okay, one thing I want to be clear about running it right now. So last year we showed the demos. I don't know, were any of you here last year or not? No one? Okay. So the demos in CellOrganizer are seeded. So you're guaranteed to get the same result every time that you run it. But these are not, meaning that even if we train on the same model, each of us should get a different image. Okay? So I'm going to import it. Select dataset, click, and it's going to take me back. And now I'll see a file called here called, it's a MAD file, it's binary data. Let's see if we can do some stuff. We retrieve information about the model file. So I'm just going to click here, retrieve information. It's going to open this menu, and it's just going to ask me to select a model. The only model I have in this history is the MMBioS. So I'm just going to click on that and click execute. And what model information it's going to do is just going to print out a very simple detail about what are the contents of the model. At this point, the only thing it's displaying is the model class and the model type. Okay? Is it done? Let's see. Watch. See those jobs that have like 20 seconds? Those are the ones that we just clicked. It's taking 30 seconds. So when this disappears, we know we could all go back. But you know what? Let's not wait. It will take like a minute. Oh, it's back. There. And it says that it returned an HTML file. Right? That's why we see the HTML here. If it's an HTML file, I can simply click on this little eye icon and it should open it.
No, no, no.
And it gave me this information about the model. It says that I trained this 2 years ago. Name is all, meaning it has protein, cell, and nucleus. Cylindrical, nuclear, 3D. Okay. It kept the full resolution at the protein level, it downsampled at the cell level. Okay, that's the information. Meaning that I should be able to synthesize an image from this. So I'm going to go to synthesis. I'm going to synthesize an option. Now, like I told you before, we only kept the 3 most important things open to you guys because it's the ones that you care. If you click synthesize, it will create an OME TIFF. If you click SVML, it clicks SVML. The OBJs are for to import into Blender and other tools. And the index image are only important for the 2D case, even though you could create a 3D index image to use with tools like Virtual Cell. Okay, so let's do something. Here it says that I can make a framework or a protein, but I'm just going to make a nucleus and a cell membrane because I want to get an image fast.
That's your favorite conversion. Nothing.
And then I'm going to click list of models, and I'm going to use this model. Did everyone get the report? Okay, so now all we need to do is click execute. All of these are set to no, execute on might, and then make sure that you only get a nucleus and a cell membrane, and click execute. But we're going to go back to synthesize an instance, and we're going to select framework and protein. We're going to go back again to might, okay? But now we're going to click yes on SVML, let's say wavefront. Is everyone there? And then we're going to click execute again.
It's just empty, I guess.
Oh, wait. So in my case, look, I click the first image that I synthesize is here. If I click on images, you'll see cell.ome. Do you not get the same results I got? I mean, we're going to get different images, but you're supposed to get one image. Okay, so so it generated an image. Now generating an image from a protein pattern, it's a little bit different because a Vesicle model has 4 components. It has texture, position, frequency, and size. And you don't have control into any of those. So if I were to look at, let's say, one of these, oh, it's actually creating a mesh. It already finished. But if you were to run it on your laptop, you would see that it would sample from the frequency model, and it would determine how many objects it needs to create. And then it will start populating the image. Now, in some of the patterns, like in some of the patterns where you have a lot of objects, the problem is that you are bound to reach some collisions. Okay, because we can only generate objects, and we can guarantee they're not going to collide, I think, at 1 standard deviation. But after that, there might be some collisions. So there's an additional check that if 2 objects overlap, we just remove the object and resample until we have the X number of objects that we sampled originally. Okay? There's a way to override the frequency model, though. You can use that in CellOrganizer, where you can say, I want to do a simple experiment like the movie that we showed this morning, or the experiment that Tom showed a while ago, where we sample from the endosomal pattern, and we said just generate 1 endosome instead of whatever it's in the model. Okay? So I'm going to go back, and if I click image here, I should see 1 OME. It's an image. It's in a TIFF format. I click, and I should get an image. Right? There's my image.
There it is in the background.
Oh, there it is. Okay. Why is the image so small? Well, because I downsample while I trained, so I produced an image that was smaller than the original resolution. Okay. In the desktop version of CellOrganizer, you're allowed to upsample if necessary. You can force the system to do that. That's especially useful when you're synthesizing from like a diffeomorphic model, but you want a high resolution, and let's say the microtubule model. You can do that. And then again, this is an OME TIFF. Now we're at the stages where this is the nuclei, and that's it. And this is the cell. Okay, so let me go back to the exercise. And according to this is still running. So we'll have to wait a little bit. But I'm going to click here to History Options. And I'm going to click on Save History.
Oh, now it's done.
And now apparently it's done for exercise 1 as well. Apparently I'm done training my model. So again, go here to the little gear. And in the gear, select Saved Histories. And if you click, it will take you to this page where you can select all of your different histories. This goes very well if you want to organize your different experiments or your different models. I'm going to click and go back to exercise 1, switch, and it took me back. So now to the right, I see 3 things. I see my initial image collection and 2 other blocks. Let's say if I click on 52, it tells me that's a MAT file. So I'm just going to click here at the little pencil, and I'm just going to edit this and say, what's this lamp, right? Lamp 2, 3, lamp 2, class 7. You can call it whatever you want. And then I'm going to go back here, do the same thing. It's telling me that it's an HTML file. I'm going to click on View Data, and basically it's giving me some information about the model that I just built. It's telling me that I downsampled the model on X and Y, and this is why I got. It's telling me that I put a cylindrical surface on the membrane. And then if you click on View Log, it will just show you all of the different steps done by the system to compute this model. And that is only for reference, so you know what happened in the background, because you don't have access to standard out here. And you can download the model, you can download the different parameters that we extracted from the model, and then you can download the options. This is really useful if you want to rerun this model in your desktop. You can submit this to the cluster, download all these things, and then retrain your model locally, just because you already have all the parameterization. Okay? Now,
So then you have to have MATLAB for this.
that is absolutely, yeah, that is correct. That is correct. You need MATLAB. So what can we do with this? Let's try to synthesize an image. Now let's try to do something more useful.
Do you always have to export it or synthesize it first without exporting, and then go back and re-synthesize with exporting? Because remember you had to choose no for everything.
Oh no, I'm just showing you different. I'm just showing you different things to do with that.
Look at the synthesis that we made in exercise 2 that should be finished now.
Yeah, it's just to show you how we do things. It's not, you don't have to do it at all. You can select whatever. So let's see, I got here, this is the image that I trained before, right? But now, if I go back to this second data set, I have more files.
Yeah.
I have, in this case, CellOME, which corresponds to the OME TIFF, and then those 3 components that are the 3 OBJ files that were generated by CellOrganizer. There's a little bug that for some reason it's considering the OBJ files as XML files. I'm trying to figure out why. So in the meantime, we cannot do any processing on this file. Well, you could download and import them into Cell Blender, of course. But within the system, we cannot do the validation. So let's do a couple of things. So I'm gonna go back to the history, and I'm gonna select, you see where it says 6 things are hidden? Okay, so I'm gonna click that, and now I'm gonna see these hidden things that were shown in my history. So now I'm gonna go to Cell OME, unhide it, and I'm gonna go to Cell and unhide it. So I'm gonna go back to the top and select hide hidden, and then everything goes away. And now in my history, I have those 2 files that were part of the images data set. Now, one thing that is very important is to validate your objects. So Tom briefly talked about validating using Meshalizer in Cell Blender. We can do the same thing here in Galaxy. So basically, if you try to validate this file, you will do it by sending it to Meshalizer, okay? And if you try to validate an SVML file, what it will do is that it will zip it and send it to the official validator in the web server of the SVML consortium, and will validate the file, and the same thing with an OME-TIFF. So if I click OME-TIFF, I should be able to see here a list of files, and the one that I care is cell1.ome, and if I click execute, it should return. It failed? Why?
How do we pull this that into this validator?
Wait, I don't know what that happened. That's shouldn't happen. Okay, so we click, in this case, validate an OME-TIFF. We want to make sure that the image that we constructed is valid. And then you'll see this menu, and then from the drop-down list, you click, and you select the object from the list that corresponds to an image, which in this case, in my case, is number 6, the one where it says Cell1 OME. I'll click it, click execute, and then it's doing the validation. And are you all getting this red?
Yeah.
That should not. Oh, look at this. This is weird. This is good. Could you make notes about these bugs that were? So in this case, look at this. It's actually telling me that it validated the file. See? It's telling me that the file got uploaded and sent to the system compared to the open microscopy schema, and it's telling me that the file is valid. But for some reason, did you see the error on top?
So
That Galaxy was restarted. I think this job is happening on a local runner. So I'm wondering that because there's so many of us trying to do the same thing at the same time. So that basically the job ran properly, it did what it's supposed to do, but it's sending an error to standard error, and that's why it's failing. So it basically compares it to the OME-TIFF schema. So it makes sure that I created a file that is consistent both in the intensity values and the header of the TIFF.
I just did it again and it went.
It worked? Yeah, I think it might be a.
So how do we get the XML files back into Wavefront OBG?
What?
Someone get error.
Yeah, that's what we're talking about. Yes?
Wavefront OBG or OBJ files. Of course, they're XML. How did you say to deal with that? Do we just change the file name or like what?
No. There's no. Oh, now it worked. The system itself has a sniffer that sort of like verifies the files, and for some reason it's not recognizing an OBJ file, but it's something that we need to figure it out at some point. But you could just click and download. It's just that it's the system itself that it's not allowing me to use the validator to validate using Meshalizer, because it's thinking it's an XML file instead of an OBJ.
So
You can still.
it actually is an XML file. So now, like, now it'd be harder to get it into Blender, right? Or
No, it didn't. It did not. No, it did not. It did not make any changes to the file itself. The system thinks it's an XML file.
My computer does too.
Yeah, so it's we'll figure it out what it is, but okay. So now, in a nutshell, you can synthesize images. You can synthesize any instance from any of our models, and you can generate the OBJ files that then you can import into CellBlender. Now, a few things that were not self-evident from this morning is the idea that we can use multiple files at the same time. The example is the following. I'm going to create exercise 3. Okay, is everyone okay? And I'm going to go back to share data, data libraries, and I'm going to try to get more models. So I'm going to import lamp, these 4. Okay? Hmm? And then include to history. So the good thing about CellOrganizer is that you could potentially mix and match all of these models. So what I'm going to do is I'm going to try to synthesize an image again. Okay? And I'm going to add a bunch of models. So let's say that the framework comes from LAMP, and then I'm just going to add the different models. So I have LAMP, DFR, Nuke, and Might. So what is going to happen in practice? CellOrganizer is going to stitch these 4 models together. It's going to take the framework model from the first model in the list, and then after it generates a framework, it's just going to populate, it's going to synthesize a protein pattern from each of the models. So I'm going to get an image that has channel number 1, nuclei, channel number 2, the cell membrane, channel number 3, it's going to be the protein from the first pattern, channel number 4 from the second, channel number 4 from, and so on. And you could potentially add as many proteins as you want. Synthesizing them all with so many objects like this will take a little bit of time. So it's basically the same thing we did before. We're just going to generate out an instance with more objects. We're going to synthesize one image from disk and like this. And this should start a process of creating a much larger image. Now what happens when you submit images at different resolutions is that it's going to select the pattern with the largest resolution, and it's going to upsample the lowers to reach the higher. Okay? So what have we done so far? We have trained a classic model with about 50 images, and we got a model back. But that model did not contain protein. So then we imported a canned model that contained a protein, and we synthesized a framework. And that framework should look similar to the one that we should have gotten a framework that is similar to a framework that would have been synthesized by the model that we trained at step 1. Then we synthesize an image with 3 channels, including a protein, and we exported that as an OMTF and as OBJ files. Okay? We validated the OMTF against the validator. We got a little bug, I think, because we were all running at the same time. We'll figure it out. But we got a valid answer. This is a good file. And then you can use Meshalyzer to get a report back from Galaxy to tell you information about the status of your mesh. And you can do that beforehand, before you import the mesh into CellBlender, so you can assess the robustness of the mesh. Then we did exercise number 3, where we're trying to synthesize with more than one model. That's fairly trivial because we're using one of our canned models. So what is the next step? Next step is jumping into the diffeomorphic model, which is the model that we talked about this morning, which I think all of you are the most interested in that model. The reason why we did the first 3 exercises is so that you could see how we can import your own images to train a model, and how can you can synthesize an instance when you get your file. Was that clear so far? Okay. So now let's try to train a model. Now the good thing about the diffeomorphic model, the 3D case at least, is that it's less sensitive to the number of images in contrast with the Vesicle model. So a Vesicle model, in our experience, we need at least 15 to 16 images to get a good model, but for to be able to get some information about the different variations in cell shape and nuclear shape, but the diffeomorphic model it doesn't need as many images. So I'm going to make exercise 4, and I just made a new history. And we're going to do the same process we did in step 1. We're going to grab some images from the repository, and we're going to train a model. Okay? The difference is that this time we're only going to select 5 images from the repo instead of 50. So I'm going to go to Data Libraries, Images, and Gila, 3D, and let's go back to LAMP. Let's go back to process, so it should be faster. And then just select 5 images, whatever images you want. I'm going to select the first 5. Are we all on the same page so far? Yep. Take a look if everyone on the same page. Is everyone, just take a look at, make sure that everyone is. Okay, and then just like before, import to history. Click selected data sets and go back. The same process we did for exercise 1. We perform multiple operations. We select all images, and we create a data set. Are we all on the same page so far? Was everyone able to create a data set list? The real issue with the diffeomorphic model is that the time it takes to compute a pairwise distance is dependent on, again, like on the resolution and the obvious differences in shape. You know, the more similar 2 shapes are, then the quicker it gets to compute the work that it takes to morph one into the other. The more separate they are, then the greater. Now, to give you an example, I've been training a model that has about 1,000 images at full resolution for about how long, Shantao? 45 days now? It's about 1,000 images. This is the one that I'm running a model to. So computing this pairwise computation can take a really long time. Okay, so that's why we're only selecting 5 images. We want to get some results today. Okay, so again, I'm going to go to training, train a generative model, and it's the same thing as before. The difference is, again, going back to the class type and model class and class type, we're going to select something different. So we're going to select the images. It is 3D. These 4 indices remain the same. The only difference is that now I'm only going to train a framework. I don't care about the protein model for now. And then I'm going to go to nucleus. And if you take a look at the table, it says that if I want to do a diffeomorphic framework 3D, my options should be the class framework and the type diffeomorphic. So the nuclear type should be diffeomorphic. And the nuclear should be what? Framework? We should repeat the same thing. What is it? Diffeomorphic. framework. And then I don't care about the protein because I'm ignoring it because I just select the framework option. And then documentation, again, put whatever you want. And then that's it, execute, and then we wait. Reality, even though we have 2 images, because we only downsample them by a factor of 5, how much is 5 choose 2? 10? Right? 5 choose 2 is 10, and it should take a couple of minutes. So training this model will take about 10 minutes, 10, 15 minutes. But we don't want to sit down and wait. No? What? No, but the problem is that the way that it works here, it's in serial. Each of these jobs is serial. So if you were to run CellOrganizer in a cluster setting, if like you were to run it directly on bridges, you could submit multiple jobs on the same data set, and then each of these computations will happen in parallel. So there are ways of speeding up this process. Okay, again, we're here, but I don't want to wait. So let's make another exercise. Let's see what a model, a diffeomorphic model looks like, a real one. You know what? Let's do the same. Let's use the same exercise, so I don't have to make another history. I'm going to go to Share Data, Data Libraries, Models. Let's see, 3D3. Is this a diffeomorphic model? Oh yeah, okay, good. Okay, and I'm going to import that to history. But I'm going to go back, and is this diffeomorphic too? Yes. So let's see what's the difference between the two. Oh, so if you go to Libraries, Models, do you know what kind of cell is this? I think it's like a, is it like a lung, something in the lung? Forget. You don't know? Yeah, I did not build this model. I think it was them. Okay, so I'm going to click and import this into my history. Remember, models H1 299 3D diffeomorphic. And I'm going to put it into exercise 4. And then I'm going to go back to my history. And now I should see my 2 jobs that are running, but I also should see those 2 models that I just imported. So this morning, Sheng Tao presented a graph about, it was a plot of the shape space. It was an embedding in 2D space. I think we could do the same thing here just to figure out what is it that we're seeing. So let's go to useful tools. Let's do information about the model. And let's click on 3D3. The 3D3 diffeomorphic model, it's kind of interesting because we didn't have any marker for the nuclei. So what Greg did, who was a former member of the lab, he sort of estimated the nuclei by the differences in intensity left at the cell channel. So if you take a look at these nuclei, they look a little bit too large, but this is sort of an estimation of what we thought, or he thought it was the nuclei. So again, this is the model. This is a model trained by Greg, 3D3, 3 years ago. So now let's try to see what it looks like. So I'm going to click on Display Shape Space Plot. I'm going to select the 3D3 model. And let's see what it looks like with the default settings. And now we wait. It's only going to make a plot. It should be very quick. Is everyone on the same page? Everyone can produce the plot? Okay, so let's do the same thing with the other model. So we're going to click on Display Shape Space Plot. We're going to select the cell and nuclear alignment, and let's see what it looks like. Now, doing this kind of embedding of the shape space into a 2D plot sort of helps you visualize how shapes, oh, there it is, it's done. How shapes look a little bit different. So I'm going to click on View Data. Oh, and this is what it looks like. So you can clearly see that there's some sort of clustering. It is not self-evident because I'm not that familiar with the contents of the model. So I don't know if this was not, this is the Jarvik, right? This is the random city tagging experiment, correct?
Yes.
So this is part of what I was talking to you before, that you were talking about how your the shape of the cell changes over time. If you cluster all of the different shapes that you see, you might get a different kind of clustering for a specific condition, right? So for example, you apply a drug and that changes the shape of the cell. And as time progresses, this shape changes. You might be able to cluster this and say, okay, all of these cells corresponds to time point 1 or condition 1. Okay, but they also look a little bit too clustered for my taste. So I'm going to go back to Useful Tools, Display Shape Space Plot. I'm going to select the 3D3 again, but let's do something like 250 and click Execute. Let's see if we could see a little bit better. Now I'm going to go back to my job 13 and let's see what this plot looks like. This is the more complete model. Oh,
There's nothing on
no, it is, but it's gonna make it. Come on. Why it's not showing anything?
Well,
Mm.
nothing we can do now.
Let me go back. Display shape space plot. There you go. You could see a little bit better. This is the previous model. In the previous display, these kind of look a little bit too clustered, but then on further inspection, you can see that even though they reside in the same space, here to the left, there are some differences that sort of become obvious. So part of the parameters that we extract are changes in direction, though we register the images when we compare the distances, right, Chantal? Right, so they're, right, so you could see how in this case they are they're thinner, but a little bit, you know, they have these little like things to the side. These are more like a cylinder-like, and over there they're a little bit thicker. So, and then again, how you decide whatever shapes you use to, you know, to build your model, are going to provide you with different. I don't know why he wasn't able to extract. That's weird. Oh well. Okay, so this is how you would extract information. Now, the algorithm that we use to construct the matrix, where we keep all of the distances, can be estimated. So if you have a good percentage of distances that you have computed so far, then you can estimate the remainder of the distances, and then it will give you a sort of incomplete model, but you can still synthesize from that model. Okay. Let's go back and let's see if we can. I was running on exercise 3. Oh, apparently I created an image. Oh, there you go. This is the image that we generated before. As you can see, because the protein, even though this is the same, it contains the same lab model that we used in the previous exercise, because in this case the protein patterns were at a higher resolution, it upsampled the lower resolution patterns to match up the higher. So you're going to see the cell, you're going to see the nuclei, and then at the end you're going to see the different protein patterns.
Where are we looking at?
This is the image that you synthesize in exercise 3. Now, keep in mind that because we're using a pseudo-random generator, our images are going to look completely different. But you should get an image. Okay? Remember, we're sampling 4 times for location, for size, and for frequency. So you will get an image that it's the same size in the C stack. Well, not really. No, not that either. But you can see here all of the different objects, all of the different vesicles that we generated. They're not overlapping, though. It looks like they are because this is just a 2D representation. But this goes back to what we were talking before. Now you don't have to make a decision of the location of your vesicles. You don't have to make a decision of the number of objects. It's all based on data. Okay? And then by selecting export this as OBJs, then you will get them in that fashion. And by selecting and exporting them as SVMLs, you also get a representation that is SVML standard. Okay? Is everyone good what we have done so far? It's 4 p.m. Should we take a small break?
Yeah.
Let's take a small break. You think, Tom? 15 minutes? And then we can come back. By the time we come back, all of the jobs that have been running, they should be done.